Ladies and gentlemen, it's 2 o'clock. We would like to start this third plenary lecture. Before starting, I was asked to give you some information about tonight, this evening, about the social dinner. There are buses waiting outside at the main entrance, and they will leave at 6:30. And only people who paid for the social dinner are supposed to go with the bus. Yeah? And not people who are interested in the old road or something. No, only the people who go to the social dinner. There will be a bus only for the people who are attending the SACCO meeting, because there is a SACCO meeting from 6:30 to about 7 o'clock. For those people, there's a bus waiting for them to bring them afterwards also to the social dinner. Okay, so we will walk, the bus will drop us, and then we will walk 1 kilometer on the Via Appia, so the old road. And maybe you can imagine that maybe that road is not really a very comfortable road, so please take shoes which are at least comfortable enough to walk about 1 hour to the place where the social dinner is, and also 1 hour back. 1 kilometer, so not 1 hour. But it's at least to stress that comfortable shoes are important, yes? 1 kilometer. Okay, so this is formal announcement. And now I would like to introduce you the speaker of today. We were discussing with the organization here in Rome, the organizers, about a list of potential keynote speakers. And then you go along all kinds of topics you want to address. And it was obvious that we wanted to at least also one keynote speaker who presents the recent developments in a very important new type of research which developed especially in the past decade. And that's the application of biomolecular research in combination with quaternary research. And then we go to this list, and then you see a number of names. And then we came across the name of Greve also. And then we said, it would be great if she could give the talk. And then we were thinking, yeah, let's ask her. And you never know, maybe she comes. And it was great that you were positively answered, she said. And we were very, very happy that you said, okay, I will do it. And for us, it's a great honor. So for those who not so familiar with Inger Greve also, she's professor at the Arctic University of Norway in Tromsø, and she's director of the Aurora Center for Arctic Ecosystem Genomics. Her research focuses on the effects of climate change and human impact on northern and alpine ecosystems, with special emphasis on plants. She generally works on long timescales, Pleistocene, Holocene, and at regional geographical scales, circumpolar, Arctic, Alpine, and particularly on post-glacial dispersal frequencies and routes in the Amphi-Atlantic region. More recent work concentrates on using ancient sedimentary DNA in the study of past ecosystem diversity, species persistence, and ecosystem buildup. It's also what I mentioned during my opening speech. There's of course, a big need to understand the current changes in the biodiversity. And it's obvious also that the type of research Inge and her group are presenting is really very important, and it's really complete the data set we need to understand the current changes, the impact of the current changes, and therefore also taking into account what we know from the past. And it's her group, and it's Inger in particular, who really provide us with a lot of new information, which is important to understand these changes. And actually, I cannot wait until you present your data, so please, may I ask you to come to the stage and present your data on Here you can see the title of the talk, An Ancient Reconstruction Revolution Using Ancient Sedimentary DNA. Inge, please, the floor is yours.
Thank you very much, Mr. President. And thank you to the organizing committee. It's a great honor to be here and present to you what we can do with sedimentary ancient DNA and how we can reconstruct past systems. And towards the end of my talk, I will also go a little bit into how we can use these reconstructions to improve our forecast of future ecosystem dynamics. So we already know a whole lot about past ecosystems. Pollen analyses have been done throughout more or less the whole world and gives a very good reconstruction of the change in vegetation. We also know a lot from macrofossils and especially bones, which gives you the distribution of animals in the past. Unfortunately, macrofossils are not preserved everywhere. You'll be lucky to find good preservation. So you can't always put these things together. Ideally, you want to know all the different species from the same sample. And that's where DNA has an advantage. Here I saw some illustrations from the DNA results from one of our sites in northern Norway. And you can see how this ecosystem moves from a marine basin. You have an isolation basin, so it moves over to it's going to be a lake with lake biota. And you see the vegetation around the site change. So you get a lot of plants and mammals, but you get also fish, birds, and some invertebrates. I wouldn't claim that we currently get a full ecosystem, but at least we're moving towards it. And we get fragments of very many different organism groups showing there is potential for many more type of organisms than we're currently doing. And of course, we are interested in how these ecosystems change in relation to climate and change in relation to human land use, because these are the big drivers of change. But a very big driver that we haven't looked so much into is, of course, the ecosystem themselves, the interaction between the species. And I'm especially interested in how the herbivores affect the plants and other way around. So you can get ancient DNA from many different types of sources, like ice cores, permafrost samples, coprolites. We prefer to work on lake sediments for sort of ecosystem reconstruction, because everything that lives within the lake or around the lake, that will deposit DNA on the bottom of the lake. And you get these layers of DNA of the different biota through time. It's of course critical that you have a good age-depth model so that you can relate your plant and animal community to a certain time point. Another advantage of DNA is it's basically from within the catchment. So you have more precise information of where this, especially for the plants, live. Pollen might be disturbed over a long distance, but here it's a local signal. So we prefer to go out and core in the winter, not because it's much cooler, but most of all because the lakes are covered by ice in the north, which gives a nice stable platform to core from. Then we open our core first when we come into the ancient DNA lab, where we extract the DNA. And there are several methods of DNA analysis, and which method we choose depends on the purpose. So the most narrow method, I would say, is capture probes, which is very good because you can have much information on a limited set of taxa. For example, just larynx or just mammals. Metabarcoding is a high throughput method which focuses on short parts of a known DNA, sort of barcodes, and it allows throughput of many samples. And it's very much used, especially for plants, but now also for mammals and other organisms. Shotgun sequencing is on target. You basically get it all, which is really nice because you get the whole ecosystem if you have reference libraries to identify it with. And actually, reference libraries is really essential for all methods. DNA is just small strings of information, just like a fingerprint. And without a reference library, you have no idea which plant or animal this is a fingerprint of. So you need reference libraries. So we have made one reference library for Norway and polar region. Our colleagues from the Alps have made one called Phylo-Alps from the Alps, Pyrenees and Carpathians. Basically what we have done is going into our herbarium. So this is where the natural history collection really are important, because you can go into them, and we took one specimen of each species that grows in Norway or the Arctic, and we did genome skimming of that. It gives the standard barcodes, which are quite long fragments of DNA used in modern DNA studies, like for evolution or identification. We have the mini barcodes, the P6 loop of the chloroplast, which is most commonly used for plants for degraded DNA. And we have the whole genome schemes, which is used for shotgun sequence data. And our data give quite good cover of the present flora of the Arctic. So you see here the different floristic regions of the Arctic, and we are on 80–90% of this current flora we cover in our reference library. Similar, Phylo-Norway gives cover for the Pyrenees, the Alps, and Carpathians. This doesn't mean that these are the only places where you can do this analysis. There are other initiatives also going on, a big one for 20,000 species in China, for example. And also, you can do DNA analysis today, and you will find something, because some of the species will be shared between regions, but also you can reanalyze your DNA data as the reference libraries improve. So, and you can get more information out of it in the future. So we can work in parallel with reference libraries and doing the ancient DNA analysis. There are around 100 studies on sedimentary ancient DNA so far. Most of them mapped in this figure I took from a review on aquatic macrophytes. And you see, the majority of them are from Arctic and Alpine regions. And these are cold regions where the DNA preservation is very good. But we have some recent results also from just a few kilometers from Rome, 450-meter altitude, Lake Mesano. And as you know, it's really warm here. So actually, you can get surprisingly good results also in warmer places. And by far, the oldest one is 2 million years old. Until recently, we thought that the theoretical maximum age of DNA would be around 1 million years old. And after that, it would be so fragmented that you couldn't identify it anymore. Here is exceptionally good the conservation conditions for the DNA, and we can actually go 2 million years old. Basically, it means that with cedi-DNA, you can more or less cover the whole Quaternary. Of course, I don't believe we get this good DNA many places, but at least it shows this potential higher than we believed just a few years ago. And what's really nice of this study, it shows a whole ecosystem. So this study was led by Kurt Kjær and Eske Willerslev in the Copenhagen lab. And they had actually had these samples laying around for 20 years, and they tried them repeated times. And as the DNA methods improved, they finally got DNA out of it. And we matched it to the Phylo-Norway reference library. And you have to be really critical when you evaluate such old DNA, because there could be contamination, there could be something. It's really unlikely that this all was right. But what convinced me was the species that we actually find. 102 genera of plants. 61% of them are found in Greenland today. And this fits really nice with, this is the onset of the Arctic. The Arctic flora didn't exist before the Quaternary. And this is a subset of the boreal flora that actually were able to survive the cooler condition. And here we have also independent evidence from pollen and macrofossils. And actually 78 of the genera are also independently found in either pollen or macro or both. So I'm really convinced that this is a true signal. The mammals we found there are the ancestor of the mammoths, black geese, reindeer, hare, and small wolves. So now we can, not only that we found the mammals, but also we can start looking into the evolution. We can do real-time evolution. We have the sequences. We have the real sequence, not just guessing what was up in the top of the evolutionary tree. We actually have the sequences of it now. So this is a big breakthrough also for studies on evolution. I wish we had studies like this from everywhere and throughout the time periods. We don't, of course, but the largest set of DNA study so far is this one, which has 74 sites, more than 500 samples, and it covers the last 50,000 years. So here we used shotgun sequencing. It was also done in the Copenhagen lab. And then first they matched only to what was available in the reference library, in the public reference libraries at that point. And I just want to illustrate how important it is that we build up these reference libraries, because we then added the Philo-Norway, chloroplast, and nuclear ribosomal DNA, which identified much more. And then we added the nuclear part also. And this really improves identification. So this is what you need, good reference libraries. And we see the change in vegetation over time. There's a variable but relative high turnover of vegetation, especially in the very early period, 50,000 to 38,000, but also in the LGM and some more recent periods. We see that the Mammoth steppe, the steppe tundra, was mainly of forbs. So there were actually more forbs than grass. And this is a little bit opposite of what pollen analyses indicate. This has also been found in previous DNA analyses. And that's partly because pollen is produced in high amounts in grasses. They are wind pollinated, whereas the forbs are mainly insect pollinated and therefore might be underrepresented in pollen studies. So, and then we see an increase in shrubs and a decline in forb and grasses towards during the LGM. And after the LGM, we see really an expansion of shrubs and also trees. So these are the major patterns, and then we have local variation, but I will not go into all details. But then we have the mammals from the same samples. And what was very important in the study was that we found more recent last survival dates of several of the megafauna than had been found based on bones or skeleton analysis, both for mammoth, for woolly rhino, bison, and horse. And this is important because one of the theories of the extinction of mammals is that when the humans arrived to the area, they rapidly killed the megafauna. But actually our data shows that humans and megafauna coexisted in the same regions for millennia. The statistical analysis show that climate, especially precipitation, affects the vegetation, which caused the extinction. Of course, there might be alternative explanations for our data, and Miller and Simpson pointed out that big bones, as these megafauna bones, can degrade very slowly. So actually you can get DNA of an individual long time after it died. We fully agree on that, and we have actually excluded one site where we suspect that to be the case. But the other sites, we think that the stratigraphy is really straightforward, and it's unlikely that this explains why we have megafauna of different species at 14 sites. Definitely this is something that we need to look further into. And I think the best place to go is to look at lake sediment cores, which has a very good age-depth model. And also, I would like to look more into the details of the plants. And currently, metabarcoding is a method that gives much higher taxonomic resolution of plants. So these are the sites that we're currently working on where we have done plant metabarcoding. We are in process of adding animal metabarcoding to all the same sites based on 16S. And I will show you some examples of plant-herbivore interaction from our sites. We will start in the polar Ural. We have a well-dated core covering the last 25,000 years. And here Charlotte Clark has done the DNA, on the Bune has done the pollen. So it's Lake Bolshoi in the northern polar Ural, a very large lake, 10 kilometers from one side to the other. And both the pollen and the DNA is very rich. We have about twice as many species on average per sample in the DNA as pollen, and it's, as I said, it's the forbs that we get here. But the pollen and the DNA shows the same change in vegetation, so it's really coherent evidence for that the vegetation change we see here is robust. And here I would like you to focus on the blue one, the Arctic Alpine species. They have a peak at 12,700 before present, calibrated, where both the abundance of species and the number of species rapidly declines. And we wanted to know, is this where the mammoth is coming in? So, I mean, so many studies have found mammoths, and really, we wanted to do the same. And our first attempt, we analyzed some samples, and when you work with DNA, you don't see the DNA, of course. So you go in the lab, do your work, no idea what you have in the sample, do the bioinformatics, still no idea, until the bioinformatic pipeline is ready and you get your result. And we were a bit surprised to see that we got a lot of worms. That was, of course, a good result by itself, because there hardly any knowledge about the past distribution of worms. And they are bioengineers. They really are important to the soil. But why did we get worms? The biomass of worms is very high. The biomass of the native mammals is very low. And we have a problem that human DNA is often co-amplified. So we had to improve our method a bit and block more of the human DNA, which allowed us to see what's there. And now we have mammoth in 7 samples from this lake, and actually 23 from the neighboring lake, and they stop exactly here. So here you see the decline in Arctic alpine plant species and the extinction of the mammoth. So this is what I would call a bottom-up process. The vegetation affects the herbivore. Let's look at the next site, Iceland. As you see, it's far into the ocean. Hardly any mammal could walk out there, except the polar fox managed to cross the sea ice. So there were no mammals there at all until the humans brought them. One of our cores from Iceland show a very early vegetation, which is typical Arctic, with small forbs, bryophytes, and there's 2 major changes here. That's one, when we have the warming in the early Holocene, and then the second major change in the vegetation is when the human arrives. And now we analyze the same core for mammals, and that's exactly where the sheep come in. So this ecosystem has been stable throughout the oscillation of climate in the Holocene. But then human brings the sheep, and you see the change to the vegetation. So this is what I will call a top-down process. The herbivore affects the vegetation. Let's move a little bit further south to the Alps. Here we have been working on 14 sites, where we have both plant and mammals. First one we focused on was Sulsevli. And my former postdoc, Sandra Garces-Pastor, who is now working in Barcelona, she presented part of this yesterday, but I think the results are so interesting that I would like to share with a broader audience. So Sulsevli is a lake just at the tree line. You see a little bit scattered tree around the lake today. And we got a very rich plant and actually also pollen record from here. 366 different species or taxa of plants. And here we could sort many of them into elevation belt. so we know what kind of climate they are adapted to. Not unexpected, we see the subalpine and alpine in the early period. Then as it gets warmer, they have a replacement toward montane and subalpine species. But then, in recent period, you have the alpines coming back and more lowland plants coming up. So you have a mixture of all these different altitudinal adapted species. And this is, if you look at the top curve, you have the peak in the DNA richness and also pollen richness. So the high richness is composed of species adapted to slightly different climate. And how is this possible? Well, here we have different indicators of human activities. We have from the DNA, we have, for example, the arable plant species. We have the sheep coming in, around 3.2. We have the cow and goat coming in. And we see this causes the change. For example, with sheep, we can look at the density of sheep, and we find this nice shaped curve where you at intermediate densities find the highest richness of plants. As soon as cow comes into the system, it has an even much higher effect. So if you do the statistics with a cow in the system, it's climate, especially precipitation, it's the cow, and it's also fires, so based on the charcoal analysis, that determines the plant richness. We wanted to see whether this is a general pattern in the Alps. So this is a top-down process where herbivore affects vegetation. So let's go to the total data set where we have plant and mammals for these 14 sites. I would love to show you the pictures of all the plants. 722 species of plants, but I think it will take too long to show all of them. So I show you only the 29 species of mammals. And you may recognize some of them as domestic mammals. You might also wonder what the camel does in the Alps. But actually, there is historical evidence for camel being at these places at this time. And we also focus on some of the larger herbivores like the ibex, chamonix, and red deer. And here you see the richness pattern for each of these 14 lakes. And if you focus now on the top two lines, the total richness and the forb richness, so the black one is the total richness, you see it's closely followed by the forb richness because most of the species are actually forbs. And you see that they have an increase either the last 4,000 years or last 2,000 years. So it is a general pattern that plant richness increases the last few millennia in the Alps. The high diversity we see in the sites in the Alps today has not been there before the humans came. If you look at the wild compared to the domestic mammals, you see the wild ones in green, the domestic ones in orange, and you see almost a complete turnover. So as soon as people bring their domestic animals into the area, the wild ones disappear. So it's a complete turnover of the mammal populations here. And this has implications for the richness. So on the top, you see the plant richness for all 14 sites combined. So you see this increasing pattern. And the statistical analysis show that climate, both temperature and precipitation, is important. Red deer has some influence, but then cow is by far the most important. And that's because of that we see this rise in the recent area, in recent time. So cow is definitely an agent for causing the high richness we see in the Alps today. But it's not the only one. So I will not give you all details, but clearly we have a peak before cow arrived. So this one is mainly due to sheep. And if you go more into details, there is also some influence of some of the native mammals on the ecosystem. But basically, it's a top-down process. It's the mammals that determine the richness of plants in these ecosystems. The last area I want to focus on is northern Fennoscandia. Here we have 10 lakes. This area is really far away from everything. It was more or less completely glaciated. That means all plants and animals would have to come after deglaciation. So we go from scratch. We go from nothing to a buildup of the complete ecosystem we have in this region today. And they could have immigrated from southeast or east, or as the ice melted, also up the western coast of Norway. It's a long distance. And of course, I assume that the plants must have arrived first, followed by the herbivores, and the last one must, of course, be the carnivores. But let's look into the details of this. So we have 10 sites representing the different ecosystem of the region today, going from the northernmost spruce forest to the northernmost pine forest, birch forest, and also a bow tree line. And you see, you go here from 0 to several hundred plant species. And it's sort of, it's a rapid rise in the beginning, but it doesn't completely level off. Species still arrive. So we have reanalyzed the same data set now with Phylo Norway, which allows us to be very certain about the identification of plants. So we have more than 300 taxa identified. And because we are so confident now that we have identified them correctly, there's also bryophytes also. Yeah, and for each of them, just like with the last observation of a megafauna, the first observation is very tricky. So if a plant appears in one sample and then is away and then appears, it's very hard to know when the exact time was when it arrived to the area. If a plant was absent in all samples for several thousand years and then suddenly it's present in every sample, then we're quite confident that that was the time it actually arrived. So for some data, we have very good estimates of the true arrival date. And then we can see how this ecosystem changed through time. Dominance of bryophytes and forbs in the beginning. Then aquatics come in very suddenly. And then you have all different types of species, so forbs, graminoids, trees and shrubs, cryptogams. And because we know the identity of the plants, every plant is a bioindicator. It grows at a certain temperature, at a certain moisture, certain nutrient level. It depends on pollinator or not. It depends on mycorrhiza or not. So you can use all of this information. And you can see what of this influence when the plants arrive. Not surprisingly, temperature optimum is the most important factor for arrival, and especially regards the early big peak in the start. But also factors related to plant establishment, like soil conditions, light requirement, and actually a lot of factors are important. So when people use only climate to model the distribution of species into the future, this actually shows there are a few more variables that should be good to include in their models. But we can also reconstruct the ecosystem. So we can get a glimpse of the past, how it was in terms of temperature, continentality, moisture, or biotic interaction like mycorrhiza and pollinator dependence. For example, now there is a concern, will pollinators and plants sort of track the climatic change, or will they be one of them lag behind the other and then cause a dispersal lag? Actually here, in the period we have most immigration, we have highest proportion of pollinator dependence. So this study does not indicate that movement of pollinator would limit movement of plants. So we have 30 traits in our data set, but we picked the 9 that we thought was most important for dispersal and establishment. And I just showed this as an example of what you can do with this type of data. You can look at the breakpoints, and you see whether the breakpoints are the same or not for the different environmental traits. You can put it all together, so you see most arrival was around 12,000, and you rapidly have an accumulation of traits. That means at 12,000, most of the micro niches were already there. You had warm, wet places, cold, wet places, etc. It was there. But the stability is not there yet. There's still a lot of variation in the traits. But this is also where you have the biggest turnover in functional diversity. So the ecosystem function changed around 12,000. Then is a short period, or short, 2 million years. It takes 2 million years before we then see a stabilization. Last 8,000 years, we see a stabilization. Even though still, as you see on the top graph, new taxa arrive. Now the ecosystem is resilient to arrival of new taxa. So this is a well-functioning ecosystem. And then we want to combine this data with the animals. We have published one of the studies for Nordvivannet, this isolation basin where I show you the drawings in my second slide. We have 42 species of animals here. And I just want to highlight the very first period when this is marine. So we get a very good idea of the marine environment at this time. Another core that we have, which is the oldest one in our region, 16,000 years ago. The geologists were a little bit wondering whether the bottom was marine or not. I think we can conclude it is marine. This means that you can also, if you have problems deciding whether your sediments are marine or terrestrial, just run some ancient DNA and you find out. And then the first detection of terrestrial mammals is not until 11,100. But then we get more or less all the mammals we have in this ecosystem today. And very important is that all the mammals arrived after the breakpoint. So the breakpoint in vegetation and in the ecosystem here was caused by climate, not by arrival of mammals. Reindeer. Of course, you have reindeer in the high Arctic, and I thought it could have been there earlier, but it wasn't. And a possible explanation for that is the glacier was still blocking this fjord in Varanger. So maybe it just were not able to cross that fjord. It's found at most of our sites, quite high numbers. And if you look at the cover of the different cores, in some lakes you have it throughout the period, others only very recently. And of course, this is a region where people have interacted with the reindeer. First for hunting and maybe keeping a few for pulling the sledges, and then later domestication. So these islands here, for example, and peninsula, maybe the humans brought the reindeer out there. Same for Kutsjärvi, which is a spruce forest, not a natural habitat for reindeer. Maybe the humans brought them there as well. And we also see an overall increase in recent time, which coincide with the domestication of reindeer. But did this change the vegetation? We just started doing the statistics here, and we find some correlation between reindeer and vegetation. For example, these samples here had no reindeer, and they're associated with typical Arctic forbs. In the other end of this multidimensional scaling plot, you see plants that are usually associated with reindeer, like the dwarf shrubs, blueberry, and dwarf birch, or birch at least. So there is some correlation between vegetation and reindeer, and I assume we will find this for all mammals. But for the carnivores, I think we were very lucky to discover wolf in the data. because they will never be in high density populations, so the chance to get the DNA of it is very low. But we find it just in the first sample or in the sample after we find reindeer. So it's very clear it closely tracks its prey, which is a reindeer. Then we also get the really small mammals, like this the gray red-backed vole, which we had in many samples. It's only found at 3 of our locations, although I think it could thrive at others. We also find it scattered first at 10,002, and then disappearing and then coming back. Same we see for gauptjern, 1 record and disappearing and coming back. We don't know whether it died out in between and recolonized or actually managed to stay somewhere and we just didn't detect it. Norwegian lemming, that's sort of a key species for Fennoscandia, because actually the total present-day distribution is within the area that was glaciated. So where does this species come from? And where did it survive the glaciation? We didn't define it until 9004. So we thought it should have been there earlier. And especially because one of the theories is that it evolved somewhere within the glacier area in small ice-free refugia. It's actually found fossil in Western Norway. So I'm not sure why we didn't find it before. We find it at many lakes, but a few of them we don't find it, even though we would expect it based on the current vegetation. So we'll have to dig a little bit deeper into this data to figure out. European beaver. It arrives exactly at the expansion of trees. You see it even clearer here, at the expansion of pine, sorbus, and populus. So here you see an animal that really can track its climatic niche or its environment. We found it at 4 of our sites. All of them are out of present-day range. And we were most surprised to find it at this site, which doesn't look as a typical habitat for beaver. But when we looked more into the details, we have trees just in the same samples as we have beaver. So it was in the early Holocene warm period, it was briefly some trees around this lake, and that was when the beaver was there. Next one to appear in this ecosystem is the elk. It actually arrived 500 years after tree expansion, which was a little bit surprising. I mean, look at those legs. Why is this one slower to get up there than the beaver? And actually, there's a ongoing study from Fennoscandia that shows that it's really rapidly responding to climate changes. So in modern time, it's rapidly responding, so rapidly that we actually have to adapt our plants for it, for where we build houses, because we don't want them in our gardens. Actually, a colleague of mine met it on the way to work one day. They do occur, and we don't want to have conflicts with that. So it's a little bit, so the paleo data here are different than what the modern studies show. But okay, it did arrive. It's a megafauna, it's huge. Could this cause the stabilization that we see in this period? It's just before we see the stable period. And it's claimed that megafauna have a major effect on vegetation. Actually, we haven't done the statistics yet, but I could say because we only found it on 3 sites, we can be sure that this is not a major driver of the stable vegetation. And the last mammal I would like to mention here is the brown bear. We only have it at 4 lakes from 5,400, so very recently, again outside the present range, and no effect on the vegetation. So, to sum up what we have learned so far, we have a bottom-up cause of where the vegetation change caused an extinction of the mammoth. We have low interaction in Fennoscandia. We have top-down effect, especially where we have domestic mammals in Iceland and in the Alps. The next step is to get a denser network of samples. We have tested a few samples from this region where I see LGM samples wanted. We have had low success of extraction of DNA there. So if anyone here have samples, I would be very happy if I could get access and just test a little bit DNA. Send me an email. So the reason I want it is because, for example, for pollen, you have reconstructed the migration of trees. I would like to do this for all organisms. And for that, we need a really dense network of sites. And then I want to use this also to start modeling the future. So it is quite complex interaction that explains the abundance, sort of the population dynamics of each individual species. We can express this mathematically. So each species has a growth rate. And this growth rate is composed of a basal growth rate, which is how well the population can grow under different climate. And you can get the climate data from independent reconstruction, from the Icelandic ice core or other data. And then you can get the data on each species' climate requirements by comparing present-day distribution with present-day climate, so what you do for niche modeling. That will give you the basal growth rate. And then this in the interaction terms. And this is where the payload data are so important, because there's no other way to get these interaction terms. So here we can put together the plants and the mammals and look at the interaction. And with more sites, we can start looking also at the dispersal. So you can take the time from you see it at site 1 to site 2, and then from 2 to 3, and you can average it. You can get the minimum dispersal rate, the maximum, and the average, and use this in your model. And then you can play around with your model to optimize the parameters. You can use inverse modeling, because you actually have, for each species, both plant and animals, you have the observed growth rate under different conditions. So you can then test different hypotheses using this data, and then find the optimal model and use that to model the future under different climatic scenarios. So with this, you can forecast future ecosystem of differences. So with this, I would like to thank my co-workers, many of them here today, too many to put all the names and institutions on this slide. I would like to thank you for your attention, and I would also like to use the opportunity to invite you to the SEDA-DNA conference in Tromsø in 2 years' time, and I can promise you that will be a cool experience.
Okay, thank you, Inge. It's really, really very good. It's really showing why we are so proud to have you here. Thank you again. Okay, see you all at the bus, or in the bus.